So next slide. Okay, next slide.
There.
All right.
Cool.
So, really what this work that we've been talking about today addresses is the number one biggest unmet need in organ transplantation now, which is that we have over 100,000 people in pink on the slide on the left side on the graph who are waiting for a life-saving organ, but in blue on the bottom, you can see that only somewhere between 25 and 30% of those patients will receive an organ in a given year. And as a result of that gap between supply and demand, up to and greater than 40% of the patients who are waiting on the list will become too sick or die before they receive an organ. So we have, in the case of kidney transplantation, a intervention that is highly effective. And what I mean by that is in comparison to the only other way to support somebody who has renal failure, which is dialysis, transplantation results in a significant increase in longevity, twice as long people live on with a transplant comparison to dialysis on average. It results in a much better quality of life, and it actually costs a fraction of what it costs to maintain somebody on dialysis. So to keep somebody on a dialysis for years, over $100,000 to the healthcare system, and to maintain somebody with a transplant is about $17,000 a year. So you've got something that works really well, that's cost effective and gives people a good quality of life, but we don't have enough of them, right? And guess what? There are 90,000 people waiting for a kidney transplant. There's 750,000 people waiting for something that they're never going to get with other than dialysis. So there's the number of people who are deemed to be candidates for a kidney transplant are a small fraction of the total number of people who are on dialysis. And so if you had an unlimited supply of organs, you could imagine that rather than just trying to serve those 90,000 people, you could probably offer organs to individuals, the 750,000 people who are not considered candidates for transplantation currently. Next slide. So, when I think about our current paradigm in transplantation, it reminds me in many ways of fossil fuel. We've done a lot of things to try to make it better and more efficient, but it's never going to be able to offer an organ to everyone who needs it. And essentially this paradigm that somebody has to die for somebody else to live will always result in an insufficient number of organs. So we need sort of the wind and the solar equivalent in organ availability, which I think of as bioartificial organs and xenotransplantation. So, and we're working actually in both of these areas. The product that we're interested in bioartificial organs involves a xenoscaffolding. So it's a pig scaffolding, and we're really focused on lungs. And we partnered with a company called United Therapeutics that decellularizes these lungs and then lays human stem cells that have been coaxed down different developmental pathways depending on the type of tissue that you're trying to replace throughout the tracheal and bronchial and alveolar segments of the lungs. And you can imagine that it's very difficult to test these organs because they're basically chimeric. They have elements of pig, and they have elements of human. So if you tried to test these in an animal model, you would probably get a xenotype of response. And then in xenografting, Brad's already given a wonderful description of how we're able to genetically manipulate pigs. And he focused on talons and CRISPR. Actually, the pig that we used in the study was made by homologous recombination quite a long time ago. That was the technology that predated these much more efficient ways of editing genes. And essentially, what you do is you take a somatic cell and you modify it, and then inject that nucleus, remove the nucleus, inject it into an oocyte. You get a fusion type of step, and then those oocytes are implanted in pseudopregnant sows. And in the case of the organ that we used, those pigs were then bred. So it wasn't, direct cloning. The editing was bred through a line of pigs. And we also chose pigs that were blood type O, so that they could be universal donors, so that they could serve as sources of organs for any human blood type, because O is the universal donor again. Next slide. So again, the question is, how do you test these pig organs? Well, for quite a long time, Kim alluded to the standard joke that xenotransplantation is just around the corner and always will be. And the reason why is we've really been stuck in the preclinical non-human primate stage for over 30 years. And for most of that time, it's been because we weren't, first of all, there were varied outcomes for a long time in terms of how long the organs survived and, to what extent they underwent rejection. But a lot of it, I think, has been just a reluctance on the part of the people doing the work and the regulators to feel that necessarily the promising results that we've been seeing really for about 5 years or so are clearly translatable, directly translatable into the human, because there's a big gap between primates and humans, obviously. And so the idea is that we sought to find an intermediate step that wasn't as much of a giant leap. Next slide. And so what we chose to do was to consider testing these organs. Really, I mean, this could be used for any kind of high stakes clinical trials where there's not a good model for testing something that we want to then use in living human beings in a first-in-human trial, to test it in someone who's already deceased. So what exactly does that mean? Well, in our society, we consider someone who's brain dead to be dead, okay? And brain death involves a lack of blood flow to the entire brain, including the brain stem. One of the brain stem functions is to initiate respiration. So if you're brain dead and you're on a ventilator being maintained, and the ventilator's turned off, in fact, this is one of the criteria for brain death, the decedent will not be able to initiate a breath, okay? Because that's part of the function of the brain stem. Heart function is not, and much of the other physiology is not part of the brain. It's really controlled, in the spinal cord. And so, or is even autonomous, within the organ, as long as it's being perfused. So when someone's brain dead, you can maintain them for a significant period of time if you ventilate them. You can maintain their physiology. So the idea is that we could test a xenograft in someone who's been declared brain dead and who can't donate organs but wanted to donate organs. And by approaching the family and obtaining consent, instead of being an organ donor, they would be essentially a whole body donor for a period of time to allow some type of breakthrough study to be done. In this case, xenotransplantation. Next slide. So what did we do in terms of vetting this and really, determining whether this was okay to do, because this is a new concept. Okay. Now, there are some similarities to current policies to this new concept. First of all, when you check the boxes, at the Motor Vehicle Bureau, or when you register to be a donor, there is a checkbox that says that your organs can be donated for research. So a significant number of organs every year that come from brain dead donors are not suitable for transplantation and will go for research studies. There's also, a very old concept of anatomical donation, where somebody can donate their body to science. It's usually to medical students or to prosection studies. And so it's not an entirely foreign concept, but it had to be really extensively studied. And so we and others brought together medical ethicists, legal experts, religious experts to all look at this concept. And you might also know that xenotransplantation has been vetted in a similar way. And you may know that the Vatican actually brought together a council in around 2000 and released a statement supporting xenotransplantation. And most of the major religions have something similar as well. Anyway, so, back to this idea of whole body donation. So we then had to, go through all the regulatory agencies that are out there and get their feeling about it. So normally we would go to the FDA, right? But the FDA basically only, and the IRB, your institutional IRB, but both of those organizations only regulate research on living humans. And since these are dead humans, they really have very limited interest. And so at our institution, we had to create a parallel organization that's called RDoc, which is now focused and has the same sort of a makeup as an IRB, but it's just for studies in the recently deceased. The FDA was only concerned about what we would do after the decedent, the ventilator was turned off in the decedent, and then what would we do with the body. The Department of Health was concerned about zoonosis. Zoonosis is obviously, something of concern that, just to define that term, that's the potential for infection between species or from animals to humans more narrowly. And so, and Brad had mentioned these porcine endogenous viruses, that has been a big concern. Although, there was a study of 160 humans that have received pig tissues or pig cells, and there's been no evidence of the porcine endogenous virus jumping out of the genome and becoming an infective virus. And that has only ever occurred in a very particular in vitro assay, which involves transformed human cells and pig cells. But it is a concern, and the Department of Health is concerned about that. And we had to actually create an operating room and an ICU in a isolation facility in order to do this work. So anyway, we went through a lot of steps in order to vet this and to create a situation where that the regulators would be happy with. Next slide. And so this all culminated, and as I mentioned earlier, in this first successful pig to human transplant. That the setup for this was that we had a donor, a local donor, who had indicated that they wanted to donate, but their organs were not suitable for standard donation. And so the family was then approached with this other avenue to donation, to fulfilling the desire of their loved one to make a gift, in this case to humanity rather than to individuals, at the time of their death. And the family consented. And we in our protocol, the ethicist had asked us how long it normally takes to allocate organs for transplantation and then procure them. And we told them that was 2 to 3 days. And they said, okay, well, that's how long we think you should, do this study, and not beyond that. So that was all, discussed with the family. We used pretty simple pig genetics. So we use a single glycan knockout. Glycan is a sugar. And as Brad said, there are some sugars whose enzymes that create the sugar have been lost in evolution from pig to human. And humans have what are termed natural antibodies, which are probably generated from exposure to these very similar glycan molecules on the surface of bacteria in our colon. So the microbiome, gives us right from birth targets that are very similar to these lost pig glycans. And so we make antibodies. We make antibodies at a very high level. Up to 1% of our total antibody, circulating antibody, is against these glycans. And those preformed antibodies have always resulted in a failure of xenotransplantation, an immediate failure, hyperacute rejection, where the kidney, rather than this nice pink kidney, would turn black within minutes after the kidney is connected to the human blood vessels. So in this kidney, what we decided to do, rather than put it where you normally put a kidney, which is in the abdomen, and you usually sew it down in the vessels, it's not where our own kidneys are. It's down in the pelvis, where there's easy access to good blood vessels. You need an artery and a vein. Artery sends blood into the kidney. The vein drains it. And then you need to connect the ureter to something. We usually connect it right to the bladder. But in this case, we decided to connect the artery and the vein, the femoral vessels. And the reason why is we wanted to look at the kidney the whole time. And we wanted to be able to biopsy it and look at it under the microscope and get those beautiful histology pictures. And then we also wanted to be able to measure the urine from the kidney itself, rather than having it dump into the bladder. And then we can't tell whether it's coming from the native ureters or the native kidneys or not. So next slide. And so this just shows you the some of the functional aspects of the pig kidney during the 54 hours that we studied it. So the orange is the creatinine. So for this individual, the scale is over on the right side. For the decedent, their creatinine was elevated. They had acute kidney injury as a result of what they went through when they became brain dead. Their creatinine was 1.8, which is elevated. A normal creatinine is usually about 1. And right after we did the transplant, which is the dotted line, you can see the creatinine immediately started coming down. And it came all the way down to 0.8. On the green in the bottom is the urine from the native kidneys, which was extremely low in the beginning, did improve in time. And then the blue line is the urine from the pig kidney. Next slide. And this is the histology that I'm sure Kim and Brad have been looking for. And I want their opinion as well, but you can see the first two panels is the pig kidney before we transplanted it, a biopsy. The middle two panels are some characteristic sections, one of a blood vessel, the other of the tubules after 6 hours from reperfusing the kidney with the human circulation. And then the last slide is on the right side. The two are the H&E, so the standard histology, and on the bottom is what's called C4D, which is basically looking at complement activation. So when an antibody binds to a target, and the targets that we're concerned about in this case are the glycans, then they will activate under the right circumstances complement, and that will come across in the C4D staining. So Brad and Kim, what are your thoughts about whether this shows any characteristics of acute antibody-mediated rejection that you would expect from preformed antibody?
So the top middle picture of that artery with muscle in the lumen, that's a common artifact of the procedure of the biopsy itself, where you get things forced around in ways that weren't present during life. So I don't think that's anything abnormal at all. That is that piece of muscle in the center of the artery is a common finding. It's a common biopsy artifact. So it certainly doesn't represent anything that threatens the graft in any way. And I think all of the common findings one would expect in acute or hyperacute rejection, I don't really see any evidence for here.
Right. So that's what our pathologist thought as well. The 54-hour C4D, they gave us 0 to 1, but everything else was by Banff criteria did not show any evidence of antibody-mediated rejection. Next slide.
We've had a lot of trouble with, getting the C4D to work well in our animal models. I think there's some inherent differences, and you can have other pathways for C4D activation, such as like the mannose binding lectin. So we know that carbohydrates and other things can probably cause some little bit of activation there too. So we've had a hard time, even in non-human primates, getting a good C, especially with immunohistochemistry, even immunofluorescence.
Well, that's a good point. I may talk to you about that offline.
Yeah. Sure.
And what we don't see though either is the other characteristic findings of antibody-mediated rejection on the histology.
Yeah.
Which would be margination, the peritubular capillaries and glomerulitis and things like that.
Yeah.
But I would like to talk to you about whether there's any possibility that these could be false negative C4D stains. So that would be, if you wouldn't mind
Sure.
talking with me about that.
Yeah.
So this is, then we stain for IgM and IgG. In xenotransplantation, we do know that there's pretty significant loads of IgM that has, in the correlate in humans that we see with allo antibody, we don't think IgM is that important. But in the xeno world, there's still this thought that it might be. So we do see some staining in the glomerulus there of IgM, but virtually no IgG staining. Now, IgM and IgG staining are difficult, immunohistology. I will admit that. But anyway, for what it's worth. Next slide. Now, one thing that I didn't mention is we use very simple genetics, but we did this interesting thing where at 2 months, we in the gal knockout pigs, we minced up the thymic lobe, one thymic lobe, and transplanted it under the capsule of the kidney of each kidney. And it's very interesting because then at 9 months, when you procure the organs, you get a fairly normal looking thymus architecture. It seems to kind of reorganize into what you would identify as, the characteristic features of a thymus. The idea here is that, so, you might ask, well, what does this study tell you and what does it not tell you? Well, I think there are two things that it tells you. One is that the kidney functioned normally in a way that seemed, like it was compatible with maintaining the function of a human kidney, in at least in the determinants that we were able to measure, in as much as it cleared the toxins, it cleared the creatinine, it made, normal, actually more than normal amounts of urine. And it also did not appear to undergo a hyperacute rejection. Beyond that, there's not a lot, just because of the time, the short time duration that we had to study the organ, that we could say. But the idea of the thymus is, many of you might know that the thymus is where our immune system is educated. And the idea of transplanting the pig thymus along with the kidney is that there are a lot of amino acid variations that are different between human and pig. And probably some of the later rejections that happen of xenografts have to do with these neoantigens. So these are things that are recognized by the human immune system as being foreign. And that recognition really is born, in the thymus. So the idea of transplanting the pig thymus is to, if you will, re-educate the human immune system to delete T cell clones that might recognize these neoantigens. And there's primate data to support this. Next slide. And so I just want to kind of end with, the next phase that we're going to do with this, the research in the recently deceased. So we're going to repeat, the work that we've done in xeno. But the next thing, and if you can just click the movie feature here at the bottom. So if you click on the slide, there you go. Yeah. So we are very close with our collaborators to having what looks like a pretty normal looking bioartificial set of lungs. And so we want to test these. Again, I told you what the challenges are in testing these in animal models. We want to test this in this same model, of the recently deceased subject who donates their body. But we'll do this in an ex vivo way and not like actually connect the blood vessels directly, but circulate the human blood through the organ while ventilating the lungs in order to look at functionality. Next slide. So as you can imagine, this takes a gigantic team to really, this was 5 years really in the making, and a huge team of really dedicated people were involved. But at the end of the day, I think, what I would really like to close with is to say that this gift that this family gave to humanity by consenting to, having their loved one participate in this study after their death is a extraordinary thing. And I really think that this will accelerate the next steps, which would be the first in living human trials. And I know we've kind of run over, and I apologize for that. And I apologize for all the difficulty in getting these slides. But I'm happy to answer any questions if we have any more time.
Yeah, no, we have time. The session formally ends at 5:20 your time, so we have lots of time. And we can go beyond that. So if you, maybe if you could stop sharing your slides and we can talk as more of a group. Let's see here. Yeah, so one question that's important to me, and I hope it's of interest to others also, is the question of whether the Banff classification, existing classification, is going to be sufficient for the world of xeno, or whether we need a whole new classification, or whether the future lies somewhere in between those two. You could think, for instance, that in a successful world of xeno, that maybe a lot of the biopsies would not show rejection of any type, but would show other things. And those other things may be somewhat pig specific, and maybe would be things not commonly seen in human to human transplants. But
I think Brad is going to be the best person to answer that. He's looked at a lot of these sections, from the primate work that he's been involved in.
right.
So Brad, if you could comment on that, that would be great. Whether, certainly what I've seen, you do see, you do see phenotypic signs of rejection in xenografts. They tend to be a little bit different than what we normally see in humans, but they look, when the kidneys are failing, they look more like a chronic rejection phenotype. But Brad, I'd love to hear you, but while you're doing that, I have to make a really quick phone call. So I'm going to just go offline, but I'll be
Sure.
listening.
Sure. Yeah, well, I'd say that, yeah, the Banff really should look at this closer in some way, either in a working group or whatever. But, I'd say we have, for example, Andrew Adams, who was here, had a really great xenotransplantation model. And, a lot of times it lasted a long time. Some of these monkeys, it was almost like I told him, he was using pig to monkey, and he was almost a victim of his success because he couldn't end the experiment because it just kept going and going. The kidney kept lasting and lasting, so you couldn't really finally end the experiment. But we saw some really bad glomerulopathies that are a little unusual. We even saw this weird iron deposition sometimes in the glomeruli, and some weird things. But the funny thing is they could have the worst glomerulopathy, but still have really good function. So I think it must be targeting different, not necessarily the typical HLA antibodies that we typically think of. It could be some minor carbohydrates. And so, some of our collaborators have tried to look at some of the carbohydrate, maybe not GAL, but another one, or other things. So they definitely get a worse pattern. And, one thing I noticed about the Banff classification that I've struggled when scoring like the non-human primates is that, Banff is mainly written for biopsies. And so if it's getting a little bit better with the newer sort of refining the borderline category. But, the problem was with the prior borderline, if you found just a little bit of tubulitis, you could call it borderline. But,
Right.
when we have the whole nephrectomy there, we could usually find a little bit. So, I think that Banff could have some newer sort of like ways to account for like scoring of animal specimens where you have the full nephrectomy that are interim, and there are definitely different features like the glomerulopathy I was telling them about, Dr. Montgomery. They get Andrew Adams' animal model, even though the function was really good, they would get really bad glomerulopathy. And so it seems like the structural damage is in a different area sometimes than your typical
Yeah.
tubulitis or, other
So
things.
my feeling is that in this 2018 paper, talking about the Banff classification of tissue engineering pathology, we were farther ahead of our time than we thought. I mean, I think that's not going to happen for quite a while, that we're going to need something like this. But I'm pretty confident that we will need at least a partial new classification when we are routinely, transplanting pig kidneys, pig organs. But it's sort of, binary. It could be that rejection is identical in the two species. Absolutely, no difference. So as long as your pathologic condition is just rejection, then you can use existing Banff classification. But I'm confident there'll be lots of other things that pig kidneys or pig organs will have that human organs just rarely or have never had, right? And so you need to sort of classify those things. And there is also, and in the course, we've used Alice in Wonderland as a metaphor for the entire 10 years of the course. And what's important there is whether compensatory hypertrophy in xenografts is going to just level off like it does with human transplants. I think there's some evidence that in some circumstances, the organs might keep growing, very, very much like the action in Alice in Wonderland, or that the amount of hypertrophy would be beyond what the recipient actually needs. They don't need the kidney to keep growing. And yeah. So I don't know whether that will occur, but there are lots of other things that you could conceive of that would require major changes in the classification just because the questions change, that if you're routinely transplanting pigs, maybe severe rejection of any type is going to be rare because of the way you sort of select the pig, genetics. But that other things that are not common in human transplants will occur. Yeah. So anyway,
Yeah. So, to the point of, this glomerulopathy, the other feature that goes along with that most investigators have seen in the primates is proteinuria, and the proteinuria can actually appear pretty early.
Mhm.
And it does seem to be associated with a podocyte injury. And again, what that is, what that represents, we don't really fully understand. The one, interesting thing is that in some of the preclinical primate work where belatacept was used, it seemed to, mitigate that proteinuria and that podocyte injury. But belatacept, just for your students, is a co-stimulatory blockade drug that is approved for human use in transplantation. And belatacept and abatacept, it's a very similar type of drug, has been shown to result in some improvement in some glomerulopathies. Again, the mechanism isn't completely understood. And then Dr. Solez mentioned this hypertrophy, which we see in these pig organs that, if you take out, if somebody donates a kidney to a loved one or to anyone, so they're left with one kidney. There is this compensatory hypertrophy that occurs. So the kidney, their remaining kidney gets bigger and can take up more work, basically. And their glomerular filtration rate actually increases to some degree after their donation, because it's cut in half initially. What we're seeing in the pig organs seems like more than that. It actually seems like the pig organ is autonomously growing like it would as if it were in a pig rather than a human, because pigs get bigger than humans, right? I mean, if you let them grow for years, and the pig kidney is about the same size as a human kidney at 9 months, but at 2 years and 3 years, it's a lot bigger. Now, if you're putting a kidney into a patient's abdomen where there's a lot of room, this probably is not going to be an issue. It has been an issue in the primate studies because the primates are small and you do get what's called a compartment syndrome. And in other words, the kidney keeps growing and it runs out of space and starts to get compressed in the primate. That probably won't happen in the human. But if you're doing a heart transplant that goes into a bony cage where there's no room for expansion, that does become a problem, or if we think it'll become a problem.
I have a question about this.
Yeah.
So aren't these organs taking a scaffold from a pig and then decellularizing it and then taking stem cells from humans? So why does it keep growing like a pig heart if those cells are from humans?
So we, remember I talked about two different things, and I kind of, maybe wasn't that clear about, bifurcating them. We talked about exactly what you're saying, the bioartificial organs, that's what they are. And then we talked about the xeno organs, which is what we did in this study that I presented to you. Oh. And then I kind of did something that I probably shouldn't have, and I jumped back to the bioartificial organs just to show that that's something that we hope to do in the next few years.
Okay.
Recently deceased. So it was a little confusing. But for the xenografts, that we just take, the genetically edited pig's kidney out and transplant it. And that kidney then will continue to grow. And so one of the knock-ins that people have made, some of the pig geneticists, so, there's knockouts, which are generally these glycans, and there are more than just the alpha-gal. The alpha-gal is the most important one, but for instance, there's one called SEMA, there's one called B4-gal, thought to be of less importance. In other words, we make less antibodies to those. But in some pigs, they've knocked out all three. And then there are knock-ins, as Brad was saying, there are transgenes that have been introduced into these pigs. And guess what? One of the most popular ones right now is to put the human growth hormone, regulator into the pig kidney. And there have also been attempts to knock out the pig regulator as well. But the idea is that, maybe we can engineer our way out of this problem, but it's probably just on a practical level not going to be as big a problem for kidneys as it will be for lungs and hearts, where you can't expand because you have a bony structure that prevents, what's happened in the primate heart transplants is the heart gets constricted by the sternum and the ribs, and it starts to go into like a restrictive cardiomyopathy, and it fails, the hearts fail. So that is a really interesting idea of what's going to, what are we going to do about this growth that we see in these pig organs?
Yeah. Okay. Well, no, I think that that's very, very exciting. And I want to just, while you're still here, see if I can. Ching made this graphic that is sort of reflecting on the inaccuracy of my timeline. You know, it's been 30 years of the BAM classification. For the last 10 years, I've been pushing tissue engineering pathology, you know, and I think to some extent I was wrong. Here we are in 2021. We don't need that classification, but for xenografts, for pigs, I think we will need something. So it's kind of interesting to think, was there a clue way back then in 2011 when in that first after dinner talk I got passionate about tissue engineering pathology?
Yeah, I think
Where
a couple.
did xenografts stand then?
Yeah, I think a couple of things have happened that are important, and I'll tell you. Some of you may know that in the 1970s there were a few attempts at using primate organs in humans. Right, the baby Faye is the most famous child that was born basically with a heart that had 3 chambers rather than 5. They transplanted a baboon heart into that baby, and the baby survived for, I can't remember exactly how long, but that wasn't a hyperacute rejection. The reason it wasn't a hyperacute rejection is that baboons don't express these glycans either. So there wasn't a preformed antibody problem. But the problem was that public opinion was never going to support the use of primate organs as the source of the xenograft. I was at a meeting in the 1990s. Jane Goodall gave a presentation at the xenograft meeting. We invited her to be the keynote speaker. At the end of that presentation, I can tell you, everybody in the room, first of all, was, teared up. And secondly, realized that at that point that we would never use primate organs. It just was a non-starter. Then a few years later, good progress is being made in preclinical primate models using pig organs. And then the whole PERV, the porcine endogenous retrovirus story, kind of hit the mainstream, and it was thought to be just a non-starter because of that. Now, it took quite a few years to assemble the right amount of information to give the field confidence again that the risk of zoonosis was probably a lot less than we thought. And we had these 160 patients that were studied who had received, porcine cells or tissues. And we were not able to ever demonstrate infection of the PERV virus through any other method other than the one I mentioned earlier. And so, eGenesis, one of the pig companies, went and knocked out all, it's maybe 60 sites in the genome where the PERV, is incorporated. But most people who have been involved in using those pigs say that those pigs aren't quite normal. And there's probably some off-target effects, when you try to CRISPR out that many genes in the genome. And the FDA sort of came around to the idea that this could be done just through surveillance. And that's what we did. Everybody who was in that room with that decedent has been monitored every month, initially it was every week, and now it's every month for PERV viruses and for a whole panel of other potential transmissible, bacteria and viruses. And the FDA thinks that that's a reasonable thing that we can do. It'll be a little more challenging when we have someone who goes home, with a pig kidney and, goes back into their normal life. But
Yeah.
we faced the same thing when people went to the moon and came back. We didn't know what they were going to come back with, So these things, I think, weighed very heavily on us. And I think probably, Kim, that was the reason that it looked like there was more advancement that was being made, in bioengineering organs. But I will say that the devil's in the details, and being involved in the bioengineered organs, it is really difficult to recapitulate certain aspects, particularly the capillaries, for some reason, are really hard to grow. And I think part of that reason is that, they don't have as clear a scaffolding, as the major components and vessels, And so for these lungs, that's been our problem. It's been the capillaries. And so there's a lot of capillary with faulty capillaries, you get leak of fluid. And, so I think you probably made a reasonable prediction, but some things changed. And then this idea of this intermediate step that we just applied, I think, as I said, I don't want to overplay what that did, but I do think there's a new energy in the field, and I think it's going to accelerate.
Right. So in that editorial in KI that has your New England Journal getting comfortable with risk article as the last reference, there is this statement from Goethe about boldness and advantages. So I think you know more about being bold than probably anybody. And where did that come from? When was it just the kind of discovery that you had inherited cardiac disease? It must have been more than that.
Yeah, I mean, I think some of your students don't know, I've had a heart transplant myself. I've had a familial, a genetic disorder. It's a single base pair substitution that results in an amino acid change in a splicing gene that regulates the assembly of the large proteins, cardiospecific proteins like titin is one. And so it causes a lot of dysregulation of the electrical part of the heart and dysrhythmias. My dad died of a sudden death. My brother at age 35 died of a sudden death. And I had many sudden deaths, but I was fortunate enough to have a defibrillator that saved my life 7 times, or had somebody nearby who knew CPR and was able to resuscitate me. But I think that, having that, wearing 2 hats, being a patient and a physician, and really understanding the patient perspective. And listen, I've done a lot of work with patients and a lot of focus groups, where we've discussed risk taking, where we've discussed xenotransplantation. And, part of that editorial was really about the patient perspective, which is that, at the end of the day, it's the patient's lives that are at stake. And I think, at least what I've gleaned from my own experience in life and from other patients is that patients are probably more willing to take risks than physicians. physicians, are, and this is particularly true in transplantation, where we make all these decisions about whether to use certain organs or not. And once we decide to use that organ, then we're responsible for what happens. If we decide not to use the organ and we decline the organ, then we're not really that responsible. I don't think we feel the same level of responsibility for the patients who are waiting on the list, because when they die on the list, it's not directly, there's not a direct line to something that we did. But the point I make in this editorial is acts of omission, I think, are just as important as commission, and that we all should take responsibility for the decisions that we don't make and the decisions that we make not to do something. And so I think patients feel that too. I mean, I think patients feel that all of this progress is too slow, and they can't really understand why, we're not able to move the science forward, more rapidly. So, I mean, I think, that is always on my mind. And I believe that that you can be bold, you can be innovative, as long as you're willing to go through the laborious effort of doing it the right way. Okay, doing it in a way where you ensure that that safety is paramount, that consent is true, and that people understand what what they're agreeing to. All of the basic principles of clinical investigation have to be done in a meticulous way. And I believe then, if you do that, you do have an ability to be more bold and to really try to move the field along in a way that people will recognize is has been done properly. They may not agree with you. If you look at some of the things that have been written about what we did, and some of the if you look at some of the YouTube videos about it, and see what the comments that are made, they're not all supportive, I can tell you that. And the emails that I've received, and life would be a lot easier to just not do any of that and not take those chances that people are going to call you an abomination and curse your family and children. And I mean, it does require a certain level of just putting yourself out there when you do things like this. But as somebody who's been laying in an ICU bed waiting for a heart transplant, knows what that's like, I feel when I wake up every morning that this is what I'm supposed to be doing. This is exactly what I must do.
Yeah. So, we met face to face at the 2019 Banff meeting. One of the strange things about that meeting is it was said there that I had some sort of communication magic. I would point out you also have that, in that you reach people very well. But in my case, a lot of it has to do with the young people that I'm working with. You've already met Ching, and like this, picture of me and my robotic dog, it's better looking than either the real me or the real robotic dog, So Ching has really had a major positive influence on my life. Similarly, one of the other students, Ishida, this graphic of what the big 6 problems of the human race are, of what does it mean, the basic goodness of human beings, what the heck is that? This I think is actually the best graphic you'll find anywhere. And then the advances in protein folding in fall 2020, you would think during the pandemic, nothing important would happen. But this is really major because protein folding is to many orders of magnitude more complicated than all the big 8 problems of the human race. All these big, messy problems of the human race are much simpler and easier to conceptualize than protein folding. So since AI solved protein folding, it can probably help us solve these big, messy problems of the human race. And yeah, so these, graphics make me a much more effective person. And so the young people doing this, are really empowering me. And there is no such thing as Kim Solez, communication magic, but there is the young people, empowerment. So that's a very important factor in my life. And
Kim, if you don't mind, actually, I wanted to thank Dr. Montgomery for your presentation. I mean, I'm a second-year resident at the University of Montreal, and it's so inspiring to see someone be that bold and to, defy all of those challenges in terms of the complex ethical and scientific issues that are related to xenotransplantation. I mean, just looking at the way you meticulously backed up everything ethically and made sure the patients were fully consenting to every procedure. I can see how complex these issues are and how your boldness backed with all of those meticulous planning. It just allows you to set new standards for science and to set a model for future innovations like this. So I'm very inspired today. Thank you so much.
Thank you. And just as your mentor said, this is not about an individual. It's my name that is being, touted out there, and I'm here with you today. But the people, and just as Kim said, it's all of those people who are supporting, this movement, this advancement, that really make the difference. And I can tell you right now, just what you described, the meticulous way of vetting this, and all of the T's crossed and the I's dotted. I didn't do that. That was done by 150 other people who work alongside me, and I could not have done this without all of those people. And the role that you're playing and your colleagues are playing with Dr. Solez is very similar. It's that kind of support that makes all the difference in the world. And you'll have your moment too, and that's the idea, that you're learning how experienced people put together teams and lead teams, but it's the team that at the end of the day accomplishes these things. It's not an individual. It's just that we've done this for a long time, and and have developed that kind of aptitude. But I thank you for what you said, and I appreciate. Kim just sent me an email and said, we're doing this. Do you want to Zoom bomb? And I was like, absolutely, I will not. And I think Brad feels the same way. Like, if it's teaching, if it's the next generation, if it's, helping you guys to, to develop your careers, I will never say no to that. I say no to a lot of things, believe me, but not that.
So since January 1973, I've surrounded myself with young people. It's sort of an odd life that I've had, but it's created wonderful opportunities for me. And a lot of the big advances or the biggest advances, which maybe aren't very big, that I personally made have come from, ideas that they had. For instance, Ching, who you're talking to now, she had the idea that there was something neuroatypical in a positive way about me, and a personality test could figure out what that was. And that turned out to be absolutely true and gave me insights into myself that were like amazing and really, really helped us. And then we were talking about multiple instances of self, and she suggested that we make coroplast or cardboard cutouts of the students, life-size cutouts. So we're the only class in the world that has life-size cutouts of the students. In the height of the pandemic, where the risk of having somebody else in your office was, way up there. I had 17 students in my office in the form of these cardboard cutouts. So I was never lonely. And that's something she set in motion. I would never have had that.
That's awesome.
idea.
I
And
love that. Do you have I want to see that photo of you lecturing to the cardboard cutouts.
You get tired of them. There's so many pictures of me and these cutouts. We have a student, Taryn Stokowski, who has a picture of herself and then 2 cardboard cutouts of her. We made a cutout of all 3. And if you go to her home now, there's so many of her, it really gets confusing.
You're going to be accused of cloning, I think, your students.
Yeah, but I mean, and so there are basically 4 students, and there will ultimately be more. Each one of them has sort of transformed a major part of what I'm doing and given me a profile that I would never, ever have without them. So
all right, well, I'm going to charge your students with the next big thing, which is I want you to go back to the BAMF process and really start to work on, now that maybe we've changed the paradigm in your mind about what's going to be happening next.
yeah,
Let's really get some clear ideas. There's a lot of tissue out there now in the primates, and now some tissue from humans. Let's really get a sense for what we're talking about, with histology and immunohistology in xenotransplantation.
yeah,
Really
no,
important.
yeah. No.
All right, well, I'm gonna have to get going, but I really,
so much.
it's been fun. And Brad, I'm gonna get back with you offline and talk about the C4D staining, and try to get some of your thoughts and maybe some help as well, in trying to make some sense out of those slides and that tissue.
Sure,
Great.
and
So-
that was an amazing talk. Thank you so much. Yeah, that was awesome what you've done, and yeah.
I thought we might get you for a few minutes, but the fact you were here for the whole thing is just a fantastic.
No, it's a good moment in time. Thank you for that. But I'm at a meeting in Florida, and I just happen to have a clear couple of hours, and
Cool.
and spending it with you guys is the best thing that I could think of.
Great. Well,
So-
thank you again.
Okay.
So much. Yeah. Cool.
We'll see you later.
Take care.
Okay.
Bye bye.